Hello everyone. Our lesson is about transport across the cell membrane. So this lesson will discuss the dynamic processes that occur in the cell through the cell membrane. Specifically, it will tackle how substances of different sizes, charges, and structures are transported in and out of the cell. At the end of this lesson, you are expected to identify the different types of transport across the cell membrane and describe the different types of passive and active transport. So, motivation question for this. Have you wondered how cells acquire nutrients from the environment and excrete wastes out of them? So I hope you will be able to answer that as we end our discussion today. So there are two general mechanisms of transport across cell membranes. There are those that we call passive transport and active transport. Passive transport is defined as the movement of substances into or out of cells down concentration gradients requiring no expenditure of energy. So concentration gradients, they provide the potential energy that drives the movement and controls its direction. Active transport uses cellular energy to move substances into or out of the cell. See, what are the different types of passive transport? So you have here simple diffusion. So uh, molecules that are of higher concentration outside the cell could diffuse freely into inside of the cell through the lipid bilayer. Okay, in theory, that is if they are able to comply the requirements for a molecule to easily pass through. So there are what we call permeability factors. So because we know that the cell membrane is selectively permeable, meaning not all substances can pass through it. So it depends on the size of the molecule, the and the charge or the polarity of the molecule. So of course, smaller molecules can easily pass through, and non-polar molecules, because the cell membrane is hydrophobic, so non-polar molecules can easily pass through. Bigger molecules cannot, so they need special proteins in order for them to pass through. But again, the concept of simple diffusion is a transport of molecules from an area of higher concentration to an area of lower concentration until an equilibrium is reached. So meaning it just goes with the flow. Now there are also transporters. Now for molecules that are polar and for bigger molecules. So say for example these are polar molecules because positive and charged, so not acidic. So what happens, it cannot easily pass through the negatively charged cell membrane, so it needs special transporter proteins in order for them to pass through. Now this is a non-specific, anyone could pass through, but there are also what we call specific transporter. Okay, so specific molecules. This one is bigger, so it cannot pass through the membrane easily. So it needs the help of a transporter protein, which is specific for this type of molecules. So example is a glucose transporter. Now, if the transport of molecules through a specific or through a concentration gradient is aided by transporter protein, the passive transport process is what we call facilitated diffusion. Okay. Now, water molecules could easily pass through the membrane. Some could pass through the membrane, but majority of the water will pass through the channel protein called aquaporins that are embedded in the cell membrane. So the process of the flow of water from an area of higher concentration to an area of lower concentration is what we call osmosis. Now the flow of water is inversely proportional to the flow of solute. So say for example, this is solute. With high concentration outside, that means there is less concentration of water. With there is low concentration of solute inside, so that means there is high concentration of water inside. Now with this effect of osmosis, we identified tonicities. So tonicity is the ability of a surrounding solution to cause a cell to gain or lose water. Now three types of solutions are identified. So there are what we call isotonic solution. So this means the solute concentration is the same as that inside the cell. So inside and outside the solute concentration is the same. So there is no net water movement across the plasma membrane. If you say hypertonic solution, there is the solute concentration is greater than the inside the cell. So outside has a higher solute concentration and lower water. So what happens is that the cell would lose water. And hypotonic solution is when the solute concentration is less than that inside the cell, and so the cell gains water. So what happens to a different type of cells under this different solution? So if the cell is in isotonic solution, meaning solute concentration is equal inside and outside the cell, animal cells are in normal state. However, plant cells are in flaccid, flaccid state. If the cells are in hypotonic solution, meaning there is higher concentration of solute inside than outside, so what happens? The net movement of water is going inside. Net movement, because there will be water that will come out and water that will come in, but more of the water is going in. That's why the net movement is going inside the cell. And so if there is so much water going inside, an animal cell with only cell membrane covering it will lyse or burst. However, plant cells are in normal in hypotonic solution, which is actually necessary for them because they need continuous influx of water, because we know that water is needed for photosynthesis. In a hypertonic solution, on the other hand, this is when solute concentration is higher outside than inside the cell. So what happens is that the water from inside will go outside. So what happens? More water are going inside, and so the animal cells will shrivel and plant cells will be plasmolized. So that means because the cell wall is rigid, it's not affected, but the plasma membrane together with the plasmoplasm, with the cytoplasm will be lysed. Okay, so these are the different effects of osmosis in different types of cells. Now let us talk about active transport. We know that active transport is when there is energy expenditure in the transport of a particular molecule or molecules. So one type of active transport is when there is use of ATP, like in sodium potassium pump. So energy of ATP is needed in order to extrude sodium and take in potassium ions by the cell. Another is through secondary active transport using ion gradient as source of energy. So uniport, okay, this is when only one type of molecule is transported. However, a uniport could also be a passive transport if it follows a concentration gradient. But if it is against a concentration gradient, but using the energy of ion gradient, that means it is already a secondary active transport. There are also what we call co-transport, symport and antiport. Symport is when an ion, because of the higher concentration of ion here, it is transported inside, and with that, a molecule is co-transported within it in the same direction. However, an antiport is when a co-transported ion and a transported molecule through the ion gradient is transported in opposite direction. Another type of active transport are exocytosis and endocytosis. So endocytosis is when the cells not take in compounds or molecules or food from outside, taking in inside. Endocytosis is when some of the molecules are that are made inside the cell are excreted outside of the cell. So uh, exocytosis, the exit of the molecules could be the one that are excreted could be cellular waste or proteins that are needed outside of the cell. Now there are different ways on how cells take in nutrients from the environment. So if it is a solid particle that is taken in by the cell, that is what we call phagocytosis. This is usually done by the phagocytes. Okay, so this is also called cell eating, phagocytosis. Now, when it is just extracellular fluid that is taken in by the cell, forming vesicles. Now, this is what we call pinocytosis, okay, or cell drinking. Now, there are specific receptors in the cell that recognize specific molecules outside. So when they bind and they are recognized by these receptors, that stimulates the endocytosis. And so this is what we call receptor mediated endocytosis, forming coated vesicles inside the cell. Okay, so those are the different types of passive and active transport mechanisms, how cells will acquire nutrients from the environment, as well as discuss and how different types of compounds and molecules are transported through the cell membrane, owing that to the selective permeability of the cell membrane. So as guide questions, here are the following guide questions. So why is it important for the cell membrane to be selectively permeable? How do you differentiate passive versus active transport? What are the different forms of passive transport? And what are the effects of different tonicities to different types of cells? So I hope this will be helpful in guiding you as you review this topic. So if you have questions, feel free to contact me through my email, or you can key in your questions and some clarifications in our Facebook Messenger group chat. Thank you, and see you again in our next lesson.